Thank you so much for introducing me. Thank you all for being here. I think most people had too many caprianas last night at the party, and so I appreciate that you guys are here with me today. And thank you to the organizers for inviting me to come. So, and I'm going to tell two stories today about the microbiota and interactions with the immune system and inflammation and disease. I'm going to start with HIV and then move to Zika virus. So this figure just as a reminder that despite having antiretroviral drugs that suppress viremia and delay progression to AIDS or any illness, and typically completely prevent AIDS, individuals who are HIV infected still have increased morbidities and mortality compared to people who are not infected with HIV. We have consistently been finding that a major reason for these increased morbidities and mortality is inflammation and effects from gastrointestinal or mucosal dysfunction. So things like gut epithelial barrier dysfunction and ensuing innate immune activation, inflammatory coagulation biomarkers, and markers of microbial translocation all have predicted mortality rates, and this is typically independent of whether people have antiretroviral therapy or not. So even when you suppress the virus with antiretroviral therapy, you still have increased inflammation, increased microbial translocation. So just to demonstrate microbial translocation and what's going on in the GI tract and how important it is for HIV, I like to use this figure. So this is some work by Jake Estes' group. And this is actually in rhesus macaques, which is the model for HIV infection. And in this figure, which hopefully isn't too hard to see, this is the colon of an uninfected, do you see the arrow? There it is, sorry about that. This is the colon of an uninfected, so a healthy rhesus macaque. And the green here is showing the tight epithelial barrier of the GI tract, which is intact, and you'd want that epithelial barrier nice and tight and intact, without breaches to prevent anything from the lumen from crossing into the lamina propria, and the lamina propria then would be here in blue. The red is a marker for E. coli, which is showing bacteria, and that bacteria is in the lumen of the GI tract, where it should be, of course. However, when you infect a monkey with SIV, which is the simian form of HIV, what happens is you start seeing breaches to this tight epithelial barrier. So where these arrows are, you can see that there's these focal breaches in the epithelial barrier, and where those breaches occur, it's allowing microbial products to translocate across that barrier due to the breaches, get into systemic circulation. And we know then that having microbial products in systemic circulation, which we call microbial translocation, is highly associated with morbidities and mortality. The other thing that I think is important to mention here is not only do you see that the microbial products are coming across, but also you can see that there's more of them. So the staining here, it says it's for E. coli, but really it's going to pick up a lot of gram-negative bacteria. And the gram-negative bacteria in the GI tract tend to be the bad bugs, and we're going to talk a lot more about the microbiome and what that means in a moment. But really the goal of my lab was to understand what the mechanisms underlying this mucosal dysfunction HIV infection is. So why do you get these breaches in the tight epithelial barrier? Why does that occur? So we've been hypothesizing for a long time that both in transmission and pathogenesis, which I'm focusing on pathogenesis for this talk, you have several overlapping mechanisms that are all associated with this triage of events that results in morbidities and mortality, including this barrier damage, inflammation, and an altered microbiome. And I'm going to talk to you about how these are intertwined. So just to take a step back, and I've been using the word microbiome, but to make sure we're on the same page and you are aware of what the microbiome is, it is microorganisms in an environment. There's 10 to 100 trillion microorganisms per person. I always like to give you the fun fact that an adult has 5 to 10 pounds of bacteria. So kilograms would be what? About, I'm very bad at math. 3 to 4 kilograms of bacteria in an adult. A lot. So there's a lot of bacteria in you. You have more bacteria cells than human cells in your body. But I also want to point out the microbiome is not just bacteria. While I'm going to focus mostly on bacteria today, it also includes viruses, fungi, protists, archaea. And when we say microbiome, that also encapsulates all the genes, metabolites, and bacterial products. So these bugs are there for a purpose. They're not just hanging out feeding on us. What they're actually doing is metabolizing the food and different things that we're bringing into our body, and that provides very important food sources for our epithelial cells to maintain that barrier. It helps with interaction with the immune system, etc. I also want to introduce you to the word dysbiosis. So dysbiosis is an imbalanced microbial community, and this can be a change in either the abundance of certain bacteria or functional changes. So not just that the bacteria that are there are changing, but that what they're doing is changing as well. There are many microbiomes. This doesn't even show all of them. So we tend to think about the GI tract microbiome, and by far the most bacteria in the body is in the colon. However, there are microbiomes all over your body. You have an oral microbiome, eye microbiome, ear microbiome, lung, skin, vaginal. And virtually every microbiome site has been associated with some sort of disease. So it's been estimated that over 90% of diseases have some sort of association with microbiome dysbiosis, whether it's hypothesized that the dysbiosis of the microbiome causes disease or vice versa, the disease might cause microbiome dysbiosis. But what is very clear is that the microbiome is very, very important for human health. In the context of HIV or Zika virus, the vaginal and gut microbiomes have become very important to play. We do a lot of work in my lab in the vaginal microbiome, and it's very important. I'm not actually going to, I don't have time to talk about that today, but I'm going to focus on the gut microbiome dysbiosis today. So here's just kind of a cartoon of what I've been telling you. So you have this tight epithelial barrier of the GI tract. You have all these homeostatic cells and cytokines, and we're going to talk a lot about this cell in particular, the neutrophil, in a moment. And hopefully you have a healthy microbiome that's going to protect and maintain that barrier and have good interactions with those immune cells to make sure that you don't have overt inflammation. However, in an HIV-infected individual, what we know or have observed is happening is you have damage to the tight epithelial barrier, like I showed you in the macaque. This allows microbial products to get across. You have this altered microbiome, and you have this change from these homeostatic cells and cytokines that are there to maintain the barrier and protect you into these very inflammatory cells and cytokines in the GI tract, and we think that this is associated with the gut damage, as I'll show you in a moment. So to give an overview of what HIV dysbiosis is during HIV infection, so what has been overall shown, and there's a lot of studies, there's conflicting results as there is in anything, but overall what we tend to see in HIV infection is that you have an increase in bad bugs. So you have increased Prevotella. Prevotella are not good because they have extremely long LPS chains, and those LPS chains are very inflammatory, and it's been shown that the longer the LPS chain is, the more inflammatory it is to cells. They also have increased proteobacteria. Now, proteobacteria include many bugs that are associated with disease. And also this is important because proteobacteria tend to be very adherent. So rather than just being a lumen, they tend to get through the mucosal barrier and really grab onto the epithelial barrier. And typically, when you find bugs that have translocated, whether you look in the liver or the lymph node or the blood, most of the bacteria that you find that translocate are proteobacteria, and probably because of that adherent nature. And then in general, we find a decrease of good bacteria. So we see loss of firmicutes. Firmicutes are important. They tend to be our probiotics. So probiotics are good bacteria that are beneficial to you. So firmicutes include things like lactobacillus. And almost every probiotic is actually based off lactobacillus. You also see loss of Bacteroides. This is very important because Bacteroides make things like short-chain fatty acids, which I'm going to talk about a lot later. In the Zika virus model, but the short-chain fatty acids, specifically butyrate, are very important for maintaining the epithelial barrier, and it's a food source or an energy source for epithelial cells. So without those short-chain fatty acids, then you're going to have increased damage to the barrier. I do want to point out something very important here, is that there was a study that came out by Bajer Paredes' group a couple years ago, and now has been confirmed by other groups, that when we talk about men who have sex with men, there tends to be this dysbiosis even prior to HIV infection. So what I just described here, when you compare men who have sex with men to men who do not partake in anal intercourse, you tend to see these same changes even prior to HIV infection. So there is some, I wouldn't say controversy, but it is unclear right now how much of this dysbiosis results in HIV transmission and exists prior to HIV infection, or how much HIV actually contributes to dysbiosis. But the field is really trying to sort that out right now, and I would say that both of them contribute. So I promised you we're going to talk more about neutrophils. So neutrophils are the very first immune cell typically to the site of infection. They're the most abundant immune cell in our body. They are very important in response to bacteria, and this is why we got very interested in neutrophils. They're very hard to study. A lot of people don't study neutrophils, and that's because you cannot freeze them. So if somebody's doing a sample with HIV-infected people in Africa or wherever it is, and they're freezing blood, you're going to lose those neutrophils, so you can't study them later. So everything you have to do with neutrophils has to be done fresh, which is why I think there's not a lot of work done, and there's not a lot of understanding. And even fresh, they're very difficult to live, and that's the point. A neutrophil shouldn't live long. You want a half-life of a neutrophil to be hours. The most people typically see is days, and that's because these are very, very inflammatory cells. So they're very important in antibacterial responses by phagocytosing, degranulation, nutracellular extracellular traps, but these cells come in and they cause all kinds of inflammation. They make antimicrobial peptides, they make proteases. Proteases literally can just go in and cut up epithelial barriers because they want to get all those cells through to the site of infection. And reactive oxygen species, or ROS, which also is very inflammatory and can kill cells around it. So there's this balance that you need where you need the neutrophils to come in and have a response and protect from infection. And that can be a protective role from HIV and from secondary infections. But then there's this other side of things where you have this tissue damage. You need those neutrophils to die and apoptose. And they have a natural high rate of apoptosis because you'd want them to have this short half-life and come in, react, and get the heck out of there so we don't cause damage. So for this study that I'm going to explain to you now, what we wanted to assess was what the role of these neutrophils in the GI tract and if the neutrophils are contributing to GI damage at all. Since we know we have these dysbiotic bacteria that could be potentially bringing in the neutrophils, we wanted to assess what's going on. So we had the study design for this is we have 3 different sites. So at the University of Washington in Seattle, Washington, Northwestern University in Chicago, Illinois, and at the University of California, San Francisco. And so we had enrolled 23 HIV-infected, ART-treated, so these were all treated people, and 25 HIV-negative people, and these were age-matched. And we took colorectal biopsies and blood, and then we got same-day flying in or same-day processing of these samples so we could look at the neutrophils in the GI tract. So just jumping right into the data, what we found is that there's an increase in the total neutrophils in the colon. So we used flow cytometry to measure the neutrophils, and this had been vaguely shown before by immunohistochemistry, just showing MPO, or myeloperoxidase, which is made by neutrophils, but it's not very specific because macrophages can use it too, or make MPO too. So this is the first time that we've ever demonstrated that there is an actual increase specifically of neutrophils in the GI tract. And we adjust this for age, race, and sex, sexual preference. So remember I told you how important that sexual preference is in the role. And once we adjust for that, we still see that there's a significant increase of neutrophils in the colon. This does not occur in the blood. The neutrophils are the same in the blood. There have been studies previously in the context of untreated infection showing changes in neutrophils in the blood, but we do not see that in the context of treated infection in the United States in these cohorts. So we wanted to understand why. Why are these neutrophils there? Are more just coming in constantly? Are they not dying? What's happening? So what we did is we measured active caspase 3. So active caspase 3 is a marker of apoptosis. So when a cell upregulates the caspase 3, then it's going to die. And typically, neutrophils have very high levels of caspase 3 because, like I keep saying, you want them to die. You want them to apoptose. However, in this case, what we actually saw was, and so I'm kind of showing you the opposite thing here, is that, sorry, my arrow is gone again. There it is. Okay, oops. What I'm showing you here is actually caspase 3 low. So I'm showing you the opposite. So these are cells that are surviving. This is and we use CD16 high as well. So when it's caspase 3 low, CD16 high, that's a cell that probably is not going to die anytime soon. Whereas if they have the opposite is when they're going into apoptosis, and what we see is that there's an increase of these surviving cells in HIV infection. So we have loss of caspase 3 in HIV infection. And again, even adjusting for age, sex, race, and sexual preference, we still see this. So we hypothesized then, so we show, so these neutrophils are surviving more in the colon, so that's why they're there. and they're accumulating. So we hypothesized that this might be due to the changes in the microbiome. So in these patients, we then went in and measured the microbiome, and we measured it directly from the colon biopsy. So we're looking at the bacteria that are actually in the colon when we're actually looking at the neutrophils. So these would be the bugs that are interacting. And the colon microbiome, by the way, looks a little bit different than the stool microbiome. So I think it's very important that we're looking directly at that mucosal site for the study. What we see is that there are slight increases. We didn't see statistical changes in the Prevotella or Lactobacillus because it's a very small study. However, when we actually look at this Lactobacillus Prevotella ratio, and this is one of the things that we're really finding in the field, is that it's very hard. So when you do 16S sequencing to look at the microbiome, all you're getting is relative abundance. You're not getting, there's this many Prevotellas and there's this many Lactobacilli. You're getting an abundance relative to one another. And what we're finding consistently in disease is that it might not just be when you look at the overall abundance of it, but when it's an abundance relative to another, because we really need these community structures in the microbiome to really interact and engage well with each other. And so when we look at this Lactobacillus Prevotella ratio, what we see is that there's a decrease in the HIV-infected people. So this means that they have more Prevotella relative to Lactobacillus in HIV infection, which is something that had been shown before. But what's important here is that when we look at the actual survival of the neutrophils, which I'm going to show in a moment even in more detail, it's associated with this Lactobacillus Prevotella ratio. So we went back into the lab then, and we said, well, can we induce this in the lab? Can we actually take a neutrophil, put it in with bacteria, and see the same effect? And I know this is a very busy graph, so I'm going to walk you through it. But so basically what we have here is so we're looking at how much active caspase-3 low, CD16 high neutrophils we have. So what we want here, sorry, that's not going. What you can see here is that when you look at the no stimulation media, or we looked at also HIV stimulation, because we also want to see is HIV causing this effect too, that you have very low levels of these cells, which means these cells are going to apoptose. Again, it's kind of counterintuitive the way these data are shown, but that's a good thing. You want lower levels, those cells are going to apoptose. However, when you put these cells in the neutrophils in with LPS, or these Prevotella species, or this is Bacteroides fragilis, Actinobacter juni, so these are all kind of bad bugs that are have been shown to be increased in HIV infection. We can see is we can induce the survival. So it's down-regulating. Somehow these bacteria, these bad bugs, are down-regulating the active caspase-3 and keeping the neutrophils from dying. So whereas when you look at the lactobacillus, it's the opposite. The lactobacillus actually even further pushes them into apoptosis. So the lactobacillus tells a neutrophil, turn off, die, you're going to cause tissue damage, so get out of here. Whereas the Prevotella says, hey, stick around, and we're causing tissue damage, is what we think is happening here. So, and then again, going back to these, and now we're not looking at this ratios, but looking just at how many of these surviving neutrophils you have relative to the Prevotella here on the left, or the lactobacillus here on the right. And what you can see is there's significant positive correlation, how many surviving cells you have and how much Prevotella, and significant negative correlation, how many surviving cells you have and how much lactobacillus. So, and again, this is back to the in vivo data. So we really think that what's happening here is you have an increase of the neutrophils in the GI tract during HIV infection, they're accumulating, and that this is associated with decreased neutrophil apoptosis. But the HIV-altered mucosal bacteria differentially affect neutrophil apoptosis, including decreased lactobacillus species and increased Prevotella, which may prolong the neutrophil lifespan in the GI in treated HIV infection. So we are trying now to further understand how the neutrophils contribute to the HIV pathogenesis and barrier damage directly, so we can maybe elucidate targets, so maybe we can go in and stop this mechanism of mucosal damage and microbial translocation by targeting these neutrophils or targeting the microbiota that are causing this. So I'm going to switch gears here from HIV to Zika virus infection. I thought being in Brazil, it was appropriate to talk about Zika today. And so probably no news to anybody that we had that first large human outbreak of Zika virus a couple years ago. Has dampened quite a bit now, of course. However, we never know when it's coming back, so I think it's still very important to understand what is going on with Zika virus and how we can better prevent it. And one of the things is there's a lot of work on Zika virus and what it's doing in the blood and pathogenesis and building models for it. However, no one had looked at the microbiome, so we were keenly interested in what's happening in the gut and the microbiome during Zika virus infection. I already showed you this figure. We know there's lots of microbiomes, but like I said, we are unaware if, when we know that the microbiome is so associated with different diseases, we're not aware if the gut microbiome is associated with the Zika virus infection or pathogenesis. Just want to go back and reintroduce you to short-chain fatty acids, which I did mention earlier. So short-chain fatty acids are the main bacterial metabolites produced through fermentation by specific colonic anaerobic bacteria, mostly Bacteroides make these. They interact with multiple signaling molecules, and they are very beneficial to the epithelial barrier and to immune homeostasis and keeping a healthy GI tract, and really play a key role in colonic health and prevention and management of disease. So short-chain fatty acids are very important, and the only way we get short-chain fatty acids in our body is by bacteria, the bacteria that metabolize food products. Two other things I want to introduce you to. First is neopterin. So neopterin is elevated in CSF, has been reported in several central nervous system, infectious diseases, and inflammatory disorders. And then there's tryptophan metabolism, and these are all associated in the same pathways. And tryptophan metabolism, so the activation of the kynurenine pathway, is known to exacerbate progression of neurodegenerative diseases. The kynurenine to tryptophan ratio has been very predictive of disease in many instances, particularly HIV. There's been a lot of work in HIV where we've shown that very high levels of inflammation are associated with disbalance in this tryptophan catabolism pathway, also associated with loss of lactobacillus in HIV. And then we know that downstream of this whole pathway, imbalances in serotonin levels within nervous tissues and the intestine have been associated with various gastrointestinal disorders and central nervous system disorders. So this whole pathway is very intertwined with neopterin, tryptophan metabolism, and serotonin. So here we hypothesize that Zika virus induced alterations in the GI microbiome in macaques, and I'm going to go over the models in a moment, resulting in increased inflammation and decreased functionality of gut microbiota as measured by short-chain fatty acids. We also hypothesize that inflammation in the periphery induces upregulation of physiologically balanced pathways in the central nervous system, such as neopterin production and tryptophan metabolism. So these are the models. Sorry, I know it's a busy slide, but I want to make sure that we go over these different models. So first we have, for the Zika virus models, at the Washington Primate Center, where I was located when these studies were done, we had a pigtail macaque model, so this is PTM. And pigtail macaques and rhesus macaques are both big models used in HIV, and there's benefits to both of them. Pigtail macaques tend to be bigger, tend to be easier animals to work with for a lot of reasons. And so what we did here was a longitudinal study with 8 animals, 4 females and 4 males, where we infected them with Zika virus and followed prior to Zika virus and then throughout infection. We followed them and took multiple tissues, and the tissues I'm going to show you here is rectal swabs for microbiome, stool where we looked at our short-chain fatty acids, and plasma and cerebrospinal fluid, or CSF, where then we looked at neopterin, the kynurenine tryptophan ratio, and serotonin. And this is all done by mass spectrometry, where the 16S sequencing is done in a MiSeq. And then we did this. We ran this study with our lab in collaboration with Deb Fuller and Michael Gale at the University of Washington. And then we wanted to confirm results that I'll show you. And we got samples from Dan Baruch's group and at Beth Israel Deaconess Medical Center and the Center for Virology and Vaccine Research, where they had been running many rhesus macaque studies to understand Zika virus there, and we wanted to look at it in a separate model. So from them, we got samples from 8 rhesus macaques longitudinally again, where we got plasma and cerebrospinal fluid from these animals to look at neopterin and kynurenine tryptophan ratios. And again, in this one, we did not have the gut samples, so we don't have the microbiome data on that one. We just looked at these inflammatory markers. And then finally, we wanted to look at human samples and see if there's any evidence of this then being predictive of human disease. And so for this, in the Galveston National Laboratory and the Global Virus Network, we got serum, and that's all they had available was serum, so we don't have CSF or gut samples from this either. But we were able to look at 20 patients either that were completely uninfected, in early after symptoms started, so 1 to 14 days after onset of symptoms, and then post-infection was over 14 days post-symptom. And then here we were also just able to look at the inflammatory markers in the plasma, the neopterin, KT ratio, and serotonin. So jumping right into the data, we looked at the microbiome, and again, this is only in the pigtail macaque model that we had samples to look at the microbiome. And these are rectal swabs, and I split the female and the male pigtail macaques up for a couple reasons. One is because there might be some intrinsic differences between the females and the males, and second, because these groups were actually run at separate times, which in hindsight was not the best study design, but everybody was doing Zika virus studies very fast, and we, they had 4 animals available, 4 females available, and we, got them going infected, and then once those were already going, then we got a new group of 4 males and got those going. So they should have been staggered, but they were separate, unfortunately, but it's a caveat of the study. So we can't tell if differences between the males and females are just because they're, housed differently and run at a different time. So I always want to be upfront with that, major caveat. So what we see here, though, is that in blue you have significant decreases, so changes in the microbiome after Zika infection, and then in red you have significant increases from baseline, so microbes that were increased in abundance. And in the females, what you see is things like Anaerococcus and Peptophenillus were decreased, whereas Streptococcus, and of course Streptococcus can be a pathogenic bacteria, and that was increased. However, in the males we see a bit more changes, so we see things like Helicobacter. Helicobacter is not a good bug, very inflammatory. Very common in monkeys, though, I should say. So the Helicobacter increased, the Streptococcus also increased, and then you have Cynovibrio and Treponema. Again, these can be pathogenic bugs that are increased, where you have the Holdemanella and the Solobacterium that are decreased, and not a lot is known about these bugs, but they haven't been associated with disease. So we think, again, we're getting this increase in these kind of pathogenic inflammatory bacteria and a loss of potentially homeostatic community-based bacteria. So we looked at short-chain fatty acids. Again, remember that these bacteria have been associated the bacteria are very important for making short-chain fatty acids. We measured acetate, propionate, butyrate, and then down here is just all of the total short-chain fatty acids. Again, this is only in the pigtail macaques because this is the only animals we had stool for to measure this. We measure these in the stool. And what you can see is that for all of these short-chain fatty acids, you have a rapid decline of the short-chain fatty acids, which is consistent through the Zika virus infection, through major symptoms, and then as the virus goes away and is cleared, the short-chain fatty acids come back. We next looked at neopterin formation. Like I said, the neopterin formation can be downstream of this chemtrip pathway and has association to the gut microbiome as well, and is highly associated with central nervous system disorders. And here now I'm showing you this from all the groups. So on top we have plasma, and then on bottom we have CSF. So we see significant increases in neopterin in the pigtail macaques, in the rhesus macaques, and in humans. So it was very interesting for us that we're seeing this consistently through from 2 models of Zika virus infection in macaques, and we see this in humans that are infected with Zika virus. And then again, we didn't have CSF from the humans, but from the both monkey studies, when we look in the CSF, we also see increased neopterin. We also looked at the canine tryptophan ratio, which is, again, is highly associated with inflammation and disease progression in multiple models, especially HIV. And again, we see the same thing where on top you see in plasma, in both monkey models of pigtail macaque and the rhesus macaque, you see an increase in the KT ratio. And we see this in humans, so confirmed in humans that we're seeing the same thing in the plasma. And again, in the CSF, we see increases, and it's never good to see inflammation in the CSF, by the way. So this was very interesting, very exciting to see that you have this increase in the KT ratio in the CSF in both models of monkey infection. So then we looked at the serotonin. Now the serotonin you don't see as robust of a signal as what we saw with the KT and the neopterin, but again the serotonin is all the way downstream and can be highly associated with neurodegeneration, and we do see slight increases in the plasma. In humans it's not significant, but it's trending towards it. And then when we look at the CSF, we also see slight increases that then return back to baseline. But again, this is not as substantial increases as what we see with the other ones. So we want to look at microbial translocation as well. So again, we thought maybe that these microbes are altering and that might be inducing the microbial translocation. We did this by looking at LPS binding protein, or LBP. This is not the most sensitive of assays, so you can see that the data are kind of all over the place, and some of the animals are lower or higher, but we do see that there's a significant increase in the microbial translocation as measured by LBP. and this was in the pigtail macaques. And then we look at the CRP2. Again, it's much more variable, but CRP is C-reactive protein, very associated with heart disease and inflammation. And we do see that there tends to be increases in some animals, but it's much more variable. But finally, and I know this is a crazy busy plot, and I'm not going to go through each one of these and try to have you understand, but I think this gives a good overall idea. So what we do here, and this is fun, we like to do this in a lot of studies where we have multiple data sets, is we actually take these different measures. So on here you have things like the CRP, soluble CD14, neopterin, the kin-trip ratio, our short-chain fatty acids, acetate, propionate, butyrate, and all of these different markers, and then we put them versus the different bacteria that are altered. And what we do is this is an R correlation plot, and basically wherever you see red, and the thinner the red is, the darker the red is, the more significant it is, there's a significant, a positive significant correlation. And when you see the blue, the smaller, the darker, there's a negative significant correlation. And so again, not trying to get you to understand these plots here or dive into the details of it, but what is important is that we really see these associations. As you start putting all the data together, you can see that all these factors, there's a lot of associations with them. So we really think that these mechanisms are intertwined. You have changes in the microbiome. The changes probably result in microbial translocation, which is giving downstream effects in this entire kin-trip pathway, leading to increased neopterin and serotonin in both the plasma and the CSF. To conclude this part, alterations in the rectal microbiota occurs in pigtail macaques very early, 3 to 7 days post-Zika virus infection, and is associated with the decrease in short-chain fatty acid production, which occurs shortly after that, as you'd expect, 7 to 14 days post-Zika virus infection. We found increase in cerebrospinal fluid biomarkers of neurological disorders, such as neopterin and serotonin, that were detected 7 to 14 days post-Zika virus infection in all models. And this included the neopterin and tryptophan metabolites. And so just to kind of summarize, we believe that the microbial dysbiosis, loss of beneficial bacterial metabolites like short-chain fatty acids, and then ensuing inflammation and immune activation may underlie both systemic and central nervous system pathogenesis in Zika virus infection. So with that, I would just like to thank everybody in my lab. Recently moved from University of Washington in Seattle to the University of Miami, Department of Pediatrics. Charlene Miller is who really headed up the Zika virus infection studies. Tiffany Hensley McBain was a graduate student with me at the University of Washington, who ran all of the neutrophil study work. The University of Miami Department of Medicine, University of Manitoba, University of Washington, and the Washington Primate Center, specifically Mike Gale and Deb Fuller, who we ran the Zika virus studies together with. UW Harborview Hospital, Ann Collier, UCSF Scope, Peter Hunt, and Northwestern University, Tom Hope, is where we got all of the gut samples from. Those were our clinical partners for that, so thanks to them, and of course my funding. And thank you all for showing up on a Saturday morning after a big party and paying attention. So happy to take questions. Thank you.
Thank you very much. It's open for questions, and please façam as perguntas no microfone para que possa ser, quem for falar em português, para que possa ser traduzido. Any question? Okay.
Bom dia. Eu sou pediatra e eu gostaria que, se você puder me dar algumas informações em relação às gestantes HIV positivas que são tratadas durante a gestação e que tem um bebê que nasce não infectado. O que a gente pode esperar do microbioma destes bebês? Eu sei que é uma pergunta difícil, mas podemos esperar uma translocação bacteriana significativa nesses bebês?
So that is a great question. So asking about if, what was it done? So the question is asking about what to expect in a baby born to a mother who has HIV, but the baby is not infected. So there is a lot of work done on this, not my specialty, but Heather Jaspin, who's at the University of Cape Town and the University of Washington, she has done a lot of work on this. And it's very interesting. So the microbiome is definitely altered. There's a lot of associations with looking at the microbiome in babies born to HIV-infected mothers. Grace Eldravandi, also at UCLA, has done a lot of work on this, where the microbiome is definitely worse in a baby who is exposed to HIV-infected mom. They definitely have more inflammation, and they definitely have worse outcomes. And then there's also things that come into play, like breast milk versus bottle fed versus mixed feeding. Like, we know that a mix of breast milk and bottle fed is worse for the microbiome and worse for inflammation. It's very interesting. And especially for many HIV-infected mothers, they're advised, at least in the US, they're advised not to breastfeed. And so formula instead of breast milk is also associated with worse microbiomes. So I think there's a series of things that can affect a baby born to an HIV-infected mom. And that microbiome is so important early on. And when the mom has increased inflammation and a bad microbiome, she's giving her children that, because the mom is who originally seeds the microbiome. So it is a problem, and it'd be nice, we talk a lot about, trying to give babies probiotics in the beginning and seeding a better microbiome. But again, not my area of expertise. This is what I know from other studies. But wonderful question and so important.
So I have one question. Me.
Hi.
Okay. Just in this Zika model, do you think this is just a human, has anybody studied other viruses? This is just a human response to a virus infection that changes the microbiome, not really a Zika, but because we have a response to Zika or to any other infection, we would modulate the microbiome itself.
So that is an excellent question, and I always talk about this with microbiomes. So we like to do these microbiome studies, and we say, microbiome changed, and it's associated with disease, so, these are interactive. But what is the cause and effect? Is the virus causing change in the microbiome? Is a bad microbiome causing changes? In this case, because we have longitudinal samples, we do see that the Zika virus is probably changing the microbiome. But like you said, is it Zika virus, or is it because you have inflammation? So now we have cause and effect of, the different pathways. Again, the first thing we see, it's nice, and this is why monkey models are nice, or animal models in general, is that we can do longitudinal. So, in this case, having the longitudinal samples, we see first the microbiome change, then we see the inflammation. So we think the microbiome change is causing the inflammation. Now, whether that's Zika virus specific, I don't know. One of the things that we did observe, and this is kind of anecdotal, but one of the things the vet staff did say afterwards was when they would go in to take colon biopsies, that they saw after Zika virus infection, they saw a lot of slowing off of the mucus, where the mucus was coming down, and the animals had more diarrhea. So maybe the Zika virus infection is causing something in that gut, and that's changing the microbiome. But that's just speculation. We don't know exactly. And it's really important to understand the cause and effect. And it's a huge problem in the microbiome field. I like to talk about this study with where they show that the microbiome can, alter weight. And people say, oh, well, this bacteria causes people to get fat. And it says, well, is it that? Or is it just the fact that obese people are eating more pizza that are feeding that bacteria? Cause and effect is really important, and it's good to bring up, because correlation is not causation, right? So all we can show here is that there's associations, and we hypothesize they're associated. Did someone else have a question?
O microbioma hoje tem uma importância grande, a gente tem visto, principalmente associado com essas alterações que você vê com infecções. Existe algum trabalho, aí é a prática, por que entender isso? Que eu posso usar em termos de alimentação com uma gestante ou com uma pessoa que tenha adquirido essas infecções, com paciente HIV, que ajude a melhorar um pouco essa inflamação? Estou falando em termos de alimentação, ou mesmo probióticos, se uma intervenção dessa faz sentido.
So, absolutely. So great question, asking if food or probiotics or how we can treat somebody who has HIV infection or a pregnant mother with HIV infection. So the short answer and the sad answer is we're not good at changing the microbiome. It's very difficult to change the microbiome. However, probiotics do seem to have a decreased inflammatory effect. I did some work in monkeys where we showed that giving SIV-infected monkeys decreased inflammation, giving them probiotics, and that was a probiotic called VSL3. It's a combination of lactobacillus and bifidobacter. That study now has led to a big clinical trial in the U.S. through the AIDS Clinical Trials Group, where they have been giving, it's the same probiotic, they trademarked under a different name called Visbiome. And that ACTG study, the AIDS Clinical Trials Group study, has been going on for a couple of years, and the data should, I believe the data will be presented at CROI this year in Seattle in March of 2019. So we'll see if that's having a beneficial effect in humans infected with HIV. And then it's, as far as food goes, dietary fibers or soluble fibers, the fiber-rich diets have always shown the biggest impact on the microbiome. So the short-chain fatty acids that I showed you are so important. They are made by fiber-eating bacteria. So the bacteria that love to eat fiber tend to be the ones making short-chain fatty acids. So increasing dietary fiber is always tends to be beneficial, but these studies are really hard, and looking at nutrition is extremely difficult. But in general, lower carbs, higher fiber tends to be better. Probiotics don't tend to hurt. There's been 2 cases, I think, of people getting bacteremia. They were children that were very immunocompromised that got bacteremia from probiotics, and it was probably a contaminated probiotic. But I would say, as a non-medical doctor, that in general probiotics tend to be beneficial. You can also get prebiotics, which are kind of food for the probiotic. They also tend to be fibers and fructooligosaccharides, and that goes back to breast milk. Breast milk is full of oligosaccharides, which are supposed to really help seed the immune system. So those are 3 options. There's fecal transplants that haven't had a beneficial effect in HIV. We did a study in monkeys. A group in San Francisco did a study in humans, and the fecal transplants really haven't had much of a benefit in the context of HIV. Great question. Unfortunately, we don't know enough how to change the microbiome yet, but I'm working on it.
Thank you very much, Nichole. E a gente vai terminar então a sessão. Nós somos uns 45 minutos. Thank you very much.
Thank you. Obrigada.